చెప్తున్నా
సార్ నేను దాంట్లో పెట్టిన ఓకేనా జాయిన్ కమ్మని చెప్పి నా క్లాస్ ఉంది సార్ హలో
Hello, sir.
Just a minute. Is it audible to you?
Yes, sir. Loud and clear.
Yes, sir. Yes, sir.
Yes, sir.
Just wait. Good evening, one and all.
Good evening, sir.
Good evening, sir.
Good evening, sir.
Is it visible to you?
Yes, sir.
sir.
Right. Oxidative phosphorylation. Last class we completed respiration stages, only aerobic respiration, right?
Yes, sir. Glycolysis, Krebs we have completed, sir. That
Right.
oxidative decarboxylation also we have completed.
Right.
Electron transport system we have not completed, sir.
Right. Now the electron transport system.
Okay, sir.
Are you clear in mitochondria structure?
Yes, sir.
Mitochondria structure clear.
Yes,
Right.
sir.
So oxidative phosphorylation means here oxidation reaction takes place, as well as phosphorylation also takes place. As a result, formation of ATP. Formation of ATP. It is carried out by two stages. That is oxidative phosphorylation has two stages. One is electron transport system. Second one, chemoosmotic pathway or chemoosmotic phosphorylation. Chemoosmotic phosphorylation. Chemoosmotic phosphorylation. Right. Chemoosmotic phosphorylation. Right, enough. So main concept here, it is the last stage of respiration stage. Last stage of respiration, especially aerobic respiration stage. During the respiration process, food is completely oxidized inside the cell. Food is completely oxidized. Actually, the earlier stages, in earlier cycle, such as the glycolysis and Krebs and connecting link, that is pyruvate decarboxylation, what happened? Oxidation takes place, but it is not completed. Partial oxidation takes place as a result formation of NADH, FADH2, right? Hello?
Yes, sir.
Right.
Yes, sir.
Now, already formed NADH2, FADH2, these are all completely oxidized. This oxidation takes place inside the cell, especially in mitochondria, matrix, and inner membrane. As a result, energy produced. So for one glucose molecules to completely oxidized, here involved Krebs glycolysis. In the first glycolysis, Krebs cycles.
Sir, oxidative decarboxylation?
Yes, that is also,
Okay, sir.
that is shortcut connecting link. So that is another name.
Okay, sir.
Connecting link. Right. So, but energy ATP is not available until NADH2, FADH2 are fully oxidized. So in glycolysis, any energy formation, of course, only two ATPs are formed. That not complete.
Yes, sir.
What about connecting link? No ATP.
Only NADH2.
Only two ATPs are formed.
Yes, sir.
But here for complete oxidation, how many ATPs are formed? Already we did see the balancing sheet.
Yes, sir. So total 30, sir.
So yes, total 30. But in some books given 36. That whatever
36.
maybe it is the most preferable is 30.
Yes, sir.
So before oxidation, no energy. The energy produced after NADH2, FADH2, after formation NADH2, FADH2. Here, another important thing is this NADH2, FADH2 cannot react directly with oxygen. But here, oxygen can react with electrons only. Can react with electron, which electrons are released from the oxidation of NADH2 and oxidation of FADH2. So here the one concept is finally full concept here. NaDH2, oxidized into 2H plus, 2H plus, 2H plus and 2 electrons. 2H plus and 2 electron. These are oxidized into 2H plus and 2 electrons. Now this 2 electrons, through the complex factors, I will explain later what are the complex factor which are arranged in inner membrane. Through the complex protein complex factor, finally this electron received by oxygen. As a result, formation of water molecule. As a result, formation of water molecule. Right? I will tell you later. I will clearly explain how the water molecule formed. But first, remember, this is the whole concept. So it may be NaDH2 or FaDH2, anyone. NaDH2 or FaDH2. After oxidized protons, 2H plus protons released, 2 electrons also released. This electron through the complex factors, totally 4 complex, actually totally 5 complex, but only 4 complex involved to transport of electron to this electron finally submit to the water molecule. Right? Is it clear first? Hello?
Yes, sir.
Yes, sir.
Right. Where is it? Right. So that in the first reaction, electron transport, what happened in electron transport here? Electron transport. Are you able to see?
Yes, sir.
Yes, sir.
Electron transport. NaDH2.
So these are disappearing.
Might be up in here. Okay. Put this and start mute. Right. Anyway, this NaDH2. NaDH2 clear? Are you able to see this one?
Yes, sir.
Yes, sir.
Red color, right?
Yes, sir.
Yes, sir.
What is oxidation? Loss of electron.
Loss of
Yeah.
yes, sir.
Yes,
So
sir.
here two electrons lost. And also two protons also. So these electron later enter into the electron transport system. After electron transport system, chemiosmotic phosphorylation takes place. This electron transport system carried out by total how many protein complexes? 4.
4 protein complexes.
Right. Remember it. 4 protein complexes. 4 protein complexes. And chemiosmotic phosphorylation carried out by last complex, that is the fifth one. Fifth complex. Fifth complex, right? Clear? So remember, in electron transport system, oxidative phosphorylation, 2 stages. Electron transport, which is carried out by
4 protein
4
complexes.
protein complex. 4 protein complex. 4 protein complex, right? Next is chemiosmotic phosphorylation, which is involved in by last complex.
5th complex.
Fifth one, right? So your NaDH2 and FADH2 are oxidized by 2 process. Already told you these are electron transport chain and chemiosmosis, right? Two compounds NaDH2 and FADH2 donate their electrons to the electron transport chain. That is the first step. Clear? And this process was discovered by Arthur and Harden. In 1906. While describing the role of phosphate in cellular fermentation. Then in early 1940, actually first of all, this Arthur and Harden to find out in anaerobic respiration, but not aerobic. Later, this oxidation of glucose and synthesis of ATP finally discovered by Hermann Kalkar. In 1941, Albert Lippmann described the stages to formation ATP energy. So this electron transport system discovered by Albert Lippmann. This electron transport system takes place in inner membrane of mitochondria. Already told you the mitochondria structure. In mitochondria, where it is? This outer membrane, right?
Membrane, yes, sir. Christie.
Yes, in membrane called
In
as
the Christie.
actually it is somewhat like this, double layered. Double layered like this.
Yes, sir.
Which is called as
Inner membrane, sir.
intermembrane. Intermembrane, right?
Intermembrane space, sir.
Right. Intermembrane space.
That intermembrane space, yes.
In this one, Matrix, right?
Matrix, sir.
Yes, sir.
So this
The
is the inner
folded-
membrane.
Yes,
Yes.
that's the inner membrane.
This is inner membrane. So the electron transport takes place this region. Clear?
Okay, sir.
Right? So now inside matrix, outside inner membrane, intermembrane space, this is inner membrane. The folding inner membrane, right? Is it clear?
Yes, sir. Yes, sir.
Right. Right. So this takes place inner membrane of mitochondria. Is it clear? Location clear?
Yes, sir.
Already told you the total number of complexes 5. Complex 1, complex 2, complex 3, complex 4, and complex 5. Complex 1 called as NADH dehydrogenase. Complex 2 called as succinate dehydrogenase. Complex 3 cytochrome C reductase. Complex 4 cytochrome C oxidase. Complex 4 ATP synthase. For 5 complex clear?
Yes, sir.
Right. Let's see the arrangement of complex 5 complexes. So this is the first complex. Are you able to see this arrow mark?
Yes, sir.
Yes, sir.
Right, okay. Actually unable to write the pen mark. That's
So
why
you can use the pen, sir. You can use the pen.
but it is immediately disappear. I using this, but
You try, sir. You can write it. You can
here
write on the
only,
whiteboard.
but whiteboard no diagram.
Sir wants
No,
to
sir.
write in this Microsoft PowerPoint itself.
Microsoft
Yes.
PowerPoint, he cannot. You can take this on a whiteboard then.
So
As a
why don't you
PDF.
copy this picture and paste it over there,
Paste
sir,
it
on
on
the
the.
whiteboard?
Yes, sir. You can take this PPT on whiteboard.
Where do you have to copy here? So no copy.
Sir, press Control V, sir.
Control C first.
C.
Copy the one what you
He
require.
has
Then
copied
Control V.
already.
Control
So
V, sir.
Control V, sir. Or we can do one thing, sir. ఆ టెక్స్టింగ్ చేసే దాంట్లోనే కంట్రోల్ వి ఒకసారి కొట్టండి. It might come.
Copy, okay. Unable to paste here. Okay, no problem. Okay, listen carefully. I will explain later in whiteboard, okay?
Okay, sir.
Yes, sir.
Right. So anyway, so don't get confused with this electron transport and photon transport. Already explained in mitochondrial structure. This is inner membrane, right? Blue color.
Yes.
Are you follow this arrow mark?
Sir,
So you are on
only we can
whiteboard.
see on whiteboard.
Okay. Now clear?
Yes, sir.
Yes, sir.
Right.
Yes, sir.
So the blue color, this is inner membrane. Inner mitochondrial membrane, right? Are you following?
Yes, sir. Yes, sir.
Right. As we discussed, this is the matrix. This side matrix. And what about outer side? This outer membrane, and this is space.
Intermembrane spaces, sir.
This is okay. It is immediately disappear, but anyway, just find out. Clear?
Yes, sir.
Intermembrane
Yes,
space.
sir. It's standing. It's staying, sir.
It is there, sir.
Okay,
It is
okay,
staying.
okay. Right. Right. So this is inner mitochondrial membrane. This is matrix. This three region clear?
Yes,
right?
sir.
Now this is NADH plus H plus with help of NADH2 dehydrogenase convert into NAD plus 2 protons, H plus 2 protons, and releases how many electrons here?
2 electrons, sir.
Right, 2 electrons. These 2 electron first received by
Complex
FMN
one.
in complex 1, actually, of course complex 1, but in complex 1 some of the protein, some of the chemicals are there. These are F, M, N, FMN. From FMN to electron transport to the ferrous sulfur, FeS. So these two present in complex 1. FMN and FeS, the both are present in complex 1. Right? Is it clear?
Yes, sir.
Right. So these complex 1, NADH plus H plus dehydrogenase, this is the first complex of electron transport chain. It is located internal in the inner membrane, which catalyzes the oxidation of matrix NADH plus H plus or NADH2. Right? As a result, release of 2 electrons, 2 protons. Here 2 protons and 2 electrons. And these 2 electrons one by one sent to the complex 1. First in complex 1 received by FMN. Next ferrous sulfur. Ferrous and sulfur. Right?
Yes, sir.
From
Yes,
these electrons sent to the phenol cycle. Here we can observe Q. Phenol cycle. Right? From phenol cycle to this electron sent to the complex 3. This is complex 3. From complex 3 to electron, the mobile carrier that is called as cytochrome C. Cytochrome C. From cytochrome C, the electron transport to the
complex 4.
Complex 4. Already told you earlier, complex 4 to electron. Finally, sent to the oxygen.
Yes, sir.
Right? This oxygen received electron. How many electron? 2 electron. Remember.
2 electrons.
2 electron and 2 protons from the matrix. 2 protons from the matrix. As a result, formation of
water molecule.
Water molecule. Right. This is one step. They remember clearly. So these 2 electron FMN, FeS first complex.
Yes, sir.
When the electron transport through the first complex, for one electron, 2 protons sent to the intermembrane membrane space. Here, how many electrons transported through the complex one?
2 electrons are getting
2
transported.
electrons.
Yes, sir.
Each time, for 1 electron, 2 protons. Another electron, 2 protons.
Super.
Totally, the formation
4.
of proton 4.
4.
Right. 4 protons formed. Clear. Next, this quinol cycle. I will explain later the quinol cycle also, but here remember the quinol cycle from the complex 1 to electron sent to the quinol cycle. From quinol cycle to the complex 3. Right?
Yes, sir.
Right. Are you able to see this? Just wait.
Yes, sir.
Q. Is it clear?
Yes, sir.
Yes, sir.
Q
Yes, sir.
plus electron. Clear?
Yes, sir.
Yes, sir.
Plus 2 protons. As a result, formation of UB quinol. UB quinol. Simple. When one electron from the quinol cycle to the complex 3, 2 protons. 2 protons from the matrix received by quinol. Received by quinol. As a result, formation of UB quinol. Actually, there is no place to write. I hope you understand. This is only Q. First, Q receive 1 electron. As a result, receive 2 protons from the matrix.
Yes, sir.
1 electron, 2 proton. As a result, formation of quinol. This is called quinol, UB quinol. After formation UB quinol, immediately this UB quinol send the electron to the, what is the next complex?
Complex 3, sir.
Complex 3, sent to the complex 3. Apart from the electron, and in UB quinol, what are present? 1 electron and another
H.
UB
Proton.
quinone. Actually, this is
Proton,
first
sir.
UB quinone. Right one, quinone. In UB quinone, no electron. First, remember in this, no electron,
Yes, sir.
no protons, right?
No proton. Yes,
Yes,
sir.
sir.
But in UB quinol, in UB quinol, what are present? Already 1 electron
And
and
proton.
how
2
many?
proton.
2
2.
protons.
2 protons are present. But after formation UB quinol, 1 electron sent to the complex
Third
3.
complex. Yes.
What about protons? And again, this proton sent into the intermembrane space. Are you follow me?
So loss of
Yes, sir.
4 protons to the space.
Yes. For 1 electron, 2 protons
Yes, sir.
into the intermembrane space. So now UB quinol, now reverse reaction. That is, in UB quinol, electron loss and proton loss. And again, it is converted into
Quinone.
quinone. Simple.
Yeah.
Quinone, electron, proton, quinol. Now these both are lost. Both are lost
sir.
means this electron sent to the complex 3 and proton sent to the
Matrix.
Intermediate
space.
space.
Okay.
Space.
Intermediate space. Yes, sir.
Now this quinol again received another electron, always pair of electron. but in complex traveled as a pair. But when reached the quinol cycle, but quinol cycles unable to accept the pair of electron. The quinol always received the electron one by one.
Okay.
First electron received, and again 2 protons received from the matrix, converted into quinol. And after converted into quinol, immediate loss of electron.
And
loss
2
of
protons.
Right, 2 protons. And again remain as a quinone.
Quinone, yes, sir.
And again receive another electron. Always remember the pair of electron released from NaDH2 or FaDH2.
Yes, sir. Yes, sir.
Right. Now,
Yes,
where
sir.
are the electrons now? Which complex?
Now complex
4.
3.
So through complex 3, when the electron transported through complex 3, how many protons transported into the intermembrane space?
4.
4.
Each electron 2, sir.
Yes.
So total 4.
Total 4.
Right.
Yeah.
So in complex 1, complex 3, 4 electrons.
4 electrons.
Sir, 4 protons. Clear?
Yes,
Now the
sir.
electron
Yes, sir.
from the complex 3 to the cytochrome C. Cytochrome C. This is cytochrome C. This electron. Cytochrome C. So cytochrome C, actually this is the mobile carrier. Cytochrome C, mobile carrier means it is transported between the complex 3 and 4. It carries the electron from complex 3 and carry forward towards the complex 4.
4.
Right? From
Yes,
the
sir.
complex 4, this electron finally received by
Oxygen.
oxygen. First one electron. Later another electron. In the same way, in complex 4 also, the pair of electron actually, how
Goes
many
one by one.
protons to be sent into the matrix?
2. 2 is being
What
sent.
happened in complex 1 and 3?
Actually, 4.
That's it. 4
sir.
is gone.
Actually 4.
Yes.
But here, how many are formed here?
2.
Only 2.
2. Where are the remaining 2?
It's forming water,
Right.
combining with oxygen.
The remaining 2 utilized for formation of water.
Water.
So oxygen, 2 electron, 2 protons, and formation in water molecules. So here two protons are formed in complex 4. This is up to complex 4 electron transport system. Electron transport. Only this is electron transport. Now osmotic phosphorylation. This is carried out by complex 4, by this one. Right? And if NaDH2 oxidation, how many protons are formed? In intermembrane space, how many protons are formed?
4.
Total.
6. 10, sir.
Total 10, sir.
Right. Enough. Total 10. Remember.
Okay, sir. So what about complex 2, sir?
I will explain.
Okay.
Now this for just compare with matrix space, matrix side and intermembrane space. Where are the proteins? Where are the protons are high? Matrix space or intermembrane space?
Intermembrane space, sir.
Actually, the natural place of these protons are the matrix place.
Matrix. Yes,
That's why these are always try to come down inside the matrix.
Yes, sir.
But it act as an impermeable membrane. The same like as a chemi-osmotic model. Photophosphorylation. Do you know photophosphorylation? There are 2 types of phosphorylation. Photophosphorylation and oxidative phosphorylation. What is photophosphorylation? What is photo?
Light, sir.
Light.
Photosynthesis maybe, sir.
Right. It takes place in photosynthesis. In light reaction, what happened? In light reaction also electron transport system.
Yes, sir.
Already explained in offline classes.
Yes, sir.
Right. It is 2 stages. Photosynthesis 1.
Photosynthesis 2.
The first photosynthesis 2 electron transported. Finally, this electron received by NADP plus and converted into NADPH2.
Yes, sir.
After travel the proton through the coupling factor F0, F1, the ATP is formed.
Yes, sir.
But recollect once the photophosphorylation. The same way oxidative phosphorylation also. So anyway, here the 10 protons are formed.
Yes, sir.
Here, this if the 10 protons pass through this complex factor.
Five.
This is rotatory head. It has rotatory head. As rotatory head, while the protons are traveled through the coupling factor. Total 3, if the total 3 protons are transported into the mitochondria, ATP formation actually in old book given for 3 electrons, 3 protons, 1 ATP formed. For 3, if the 3 protons transported through the coupling factor 5, 1 ATP found for 3 protons. But a total 10. So in 10, if traveled through the coupling factor, how many ATPs are formed?
3 ATPs, sir.
3 ATP. That is an old one. But in new one, instead of 3, 2.5 ATPs are formed. 2.5 ATP.
Okay.
Already balancing sheet also completed. 2.5 point, right? 2.5. 2.5 ATP formed with help of ADP phosphate. Clear? This
Yes,
NADH only in pyruvate decarboxylation and Krebs NADH2. That means complex 1 started. The complex 1 started not glycolysis NADH2. The complex 1 started not glycolysis NADH2. This complex 1 started with help of the pyruvate decarboxylation and Krebs cycle NADH2. But what about glycolysis NADH2? In glycolysis also NADH2 formed, right?
Yes, sir. 2 NADH2 has formed.
Right. 2. Let me change color. NADH2. This is G for glycolysis. Glycolysis NADH2 or Krebs FADH2.
Yes, sir.
Krebs FADH2. Krebs FADH2. Oxidation start with complex 2, rather complex 1. Remember.
Okay, sir.
So directly, skip the complex 1. Glycolysis NADH2 and Krebs FADH2 both are skip complex 1. The directly from complex 2, the electrons after oxidation, here the enzyme involved in succinate dehydrogenase. Already told you the complex 2, another name succinate dehydrogenase.
Yes, sir.
Complex. So anyway, after oxidation of NADH2 or FADH2, same like here, is converted into NAD and also formation of 2 protons and 2 electrons, right? And
Yes,
same
sir.
way, this electron transported to the quinol cycle. But in what happened in the complex 1? The complex 1, when the electrons are released, it produces high energy. And also the complex 1 has proton pumping complex also had extra. That's why in complex 1, to pumping electrons, the release energy, these energy utilized for the pumping of protons from matrix towards the
Intermembrane space.
But in complex 2 has low energy and there is no protein pumping. There is no protein pumping factor. But this protein pumping factor present in complex 1 only. But in complex 2, no protein pumping factor. And when released 2 electron from glycolysis NADH2 and Krebs FADH2, low energy produced. Low energy produced because here glycolysis NADH2 present in cytosol.
Yes,
Where is
sir.
the glycolysis?
In the cytosol
Glycolysis
only,
takes place,
sir.
cytosol, but not in
Cytosol.
mitochondria.
Yes, sir.
And very interestingly, the succinate dehydrogenase also synthesis in cytosol. Synthesis in cytosol and transported into mitochondria. But this activity actively in cytosol.
Cytosol.
Right. That's why these both are low energy. When oxidized, produces low energy. Due to this low energy and lack of proton pumping factors, no protons pump into the intermembrane space. Is it clear?
Okay, sir.
So when compared with complex 1, in complex 1, clearly when the electron towards FMN, FMN2, FES, FES2
Placed into
quinol
the
cycle. When
intermembrane.
towards the quinol cycle, automatically before reach the quinol cycle, here 4 protons pump towards the intermembrane space. But in this, to pumping the protons, here involved high energy of NADH plus H plus and proton pumping factor. These both are absent in complex 2. That's why no one proton pumping towards the
Intermembrane space.
Intermembrane space. Right. The rest of the stages, that is complex 3 to cytochrome C to complex 4, the all are same. Same means how many protons are pumping through quinol cycle? This is quinol cycle.
Yes, sir.
How many protons pumped per pair of electrons?
4, sir. For a
Right.
pair of electrons, 4.
Right, 4. Right. What about in complex? Complex 4?
2, sir. Another 2 for the...
How many protons are formed? If the glycolysis NADH2 or
6 only, sir.
6, only 6. Or FADH2, like Krebs FADH2, only 6. Right?
Yes, sir.
The same, if these 6 protons through the complex factor F0, this is F0 and this is F1. This one, adhesion. While the protons are transported through the F1, this adhesion rotated. This, as it rotates, produce the energy. Now this ADP with phosphate converted into ATP.
ATP.
So here, how many ATPs are formed?
2 ATPs, sir. If we remove the complex one.
Actually 2, but according to the new version, only 1.5 ATP are formed. This 1.5 ATP for only which NADH2? Hello?
Sir.
Which NADH2? This 1.5 ATP formation. Krebs
For
NADH2,
Krebs and
glycolysis NADH2.
glycolysis.
Glycolysis NADH2.
Yes, sir.
And
Krebs
FADH2 also.
Yes, sir.
Now clear, actually the last class we discussed about the balancing sheet. Right?
Yes, sir.
Should I explain?
Total.
Should I explain again? Balancing sheet. Already clear, just open your book. The page number?
235, sir.
Rest 235 clearly given. The glycolysis, oxidative pyruvate decarboxylation, citric cycle.
Citric acid cycle.
So I already told you how many NADH2 formed in glycolysis?
2, sir.
2. So for one NADH2 enter into the oxidative phosphorylation, how many ATPs are formed?
2 ATPs, sir.
According to Tej and Jaigar,
1.5, sir.
1.5.
Yes, sir.
So total in glycolysis, three ATPs are formed.
Yes, sir.
And by substrate level phosphorylation, 2 ATP. Right?
Yes, sir. Yes,
Total
sir.
number of ATP in glycolysis, 5 only.
Yes, sir.
In oxidative phosphorylation, no ATPs.
Only 2 NADH2, sir.
Only 2 NADH2.
Yes,
If
sir.
oxidative pyruvate NADH2 enter into the electron transport system, how many ATPs are formed for one NADH2?
2.5, sir.
Right,
According to the book.
2.5. But here the how many NADH2 are formed in oxidative pyruvate?
2, sir.
2. 2 into 2.5. Total ATP number.
5.
Glycolysis ATP number 5 and oxidative pyruvate ATP number 5, right?
5. Yes, sir.
And last cycle, citric acid cycle. Citric acid cycle, 2 substrate level phosphorylation. That means
Here 6.
direct formation of 2 ATP.
Sir.
And how many NADH2 are formed in citric acid?
6, sir. 6
6
NADH2.
NADH2 formed. For one NADH of Krebs cycle enter into the electron transport, how many
Same,
ATPs
sir.
are produced?
2.5.
2.5 for 1.
1.
So your total formed 6.
6.
6 into 2.5, 15
15.
ATP. The last one FADH2, 2 FADH2.
Yes, sir.
For
Two.
one FADH2 like a glycolysis NADH2, how many ATPs are formed?
1.5.
1.5. Here, how many FADH2 are formed in Krebs cycle?
3.
2, sir.
2.
2 only.
2. 2. 2
Right,
into
2 into
1.5.
2.1,
3, sir. Total.
3.
3 ATP more.
3 ATP. Total 30 ATPs are formed.
30.
So
Yes,
this is ATP balancing sheet.
yes, sir.
All right. Electron transport clear?
Yes, sir.
Yes, sir.
Now, actually, aerobic respiration completed. Anaerobic respiration, right?
Yes, sir.
Yes, sir.
anaerobic respiration, so this is by two methods. One is fermentation, there is alcoholic fermentation. Second, lactic acid fermentation. Lactic acid fermentation. Lactic acid fermentation, right. First, let's see the alcohol fermentation. Do you know the formation of alcohol? Hello? Alcohol fermentation. Hello?
Yes, sir.
Yes, sir.
That's right. What is the substrate of alcohol fermentation?
Pyruvic acid.
Where the pyruvic acid formed?
In the glycolysis, sir.
I am asking about substrate, not end product. Substrate,
Glucose, no, sir.
right? Glucose. You're also saying glucose is the substrate for aerobic or anaerobic, right? Alcohol fermentation, first discovered by Gilosok. Alcoholic fermentation, first glucose substrate, right? What is the end product of glucose? From glucose,
We'll get pyruvate, sir.
Lactic acid formation, I think so.
pyruvate formed, right? After formation pyruvate, this pyruvate converted into acetaldehyde. By pyruvic dehydrogenase enzyme. Here the enzyme is pyruvic dehydrogenase, and as a result, production of carbon dioxide. Production of carbon dioxide. Right. Now this is the first step. In second step, this acetaldehyde formula CH3. Let me write here. Acetaldehyde reduced into ethanol, alcohol. NaDH2, NaDH2, ethanol. It's clear?
Yes, sir.
Just wait. Okay, right.
Ethanol, sir.
Okay, I will correct it. Started NaDH2. NaDH2, ethanol, right? So this is alcohol fermentation. In alcohol fermentation, first glucose is the substrate. Same glucose glycolysis. End product pyruvic acid formed. Pyruvic acid, after formation pyruvic acid, from the pyruvic acid with help of the pyruvate decarboxylase.
Dehydrogenase.
Here involved decarboxylase enzyme, decarboxylase, involved by decarboxylase. Decarboxylase formed as a acetaldehyde, release carbon dioxide. In second step, this acetaldehyde reduced into ethanol. So here this NAD reduced means hydrogens gives to the acetaldehyde. As a result, after loss, these two hydrogens convert into NAD. Right? Is it clear? Hello?
Yes, sir.
Yes, sir.
Right. Now second one, lactic acid fermentation. Right. Same in lactic acid also same. First glucose. Glucose. Lactic acid dehydrogenase converted into lactic acid. Here no decarboxylation reaction. Directly dehydrogenase. Dehydrogenase, that is reduction division. Finally, it is converted into lactic acid. Mainly this lactic acid, this is mainly takes place inside the cells, animal cells especially. what happened if you exhausted? Hello?
Anaerobic
We have
respiration occurs, sir.
not sufficient oxygen, level of oxygen. When you extremely exhausted, what happened? Or you feel like a tired,
Tired and muscles starts paining.
right? So this is happening with help of deposition of lactic acid. In normal situation, aerobic respiration takes place and it gives energy. But in overload, over exercise, what happened here? Is there any ATPs are produced? Hello?
No,
No,
sir.
sir.
In this too,
No,
of course,
no
ATPs produced, no doubt. But when compared with an aerobic, very less ATPs are produced. The ATP means here, as is glycolysis takes place in both fermentation, alcoholic fermentation glycolysis, lactic
Lactic
acid
acid
glycolysis.
fermentation.
But is there
Yes.
any pyruvate decarboxylation process in this
No,
both?
no, sir.
Any Krebs cycle in this both?
No, sir.
No Krebs cycle, no decarboxylation, pyruvate decarboxylation. So only less NADH2 are formed. Very less NADH2 are formed. That is only formed in glycolysis. But what happened here? The remaining NADH2 utilized to formation of alcohol.
Ethanol. Okay. Yes,
And
sir.
here also utilized. Here also same. NADH2 converted into NAD. No oxidative NADH2 was formed. So ATP generated very less when compared with aerobic. That's why no energy. That's why you feel like a very tired and muscle must be pain. Right? Is it clear? Hello?
Yes, sir.
Yes.
Right.
Sir, you have to use the tool, sir, to remove that marks.
Tool. Oh, just wait.
Like erase tool will be there, na, sir?
Okay. Let's take the new board. Right. And next, what is the next chapter? Nitrogen metabolism. Right. Nitrogen metabolism. Page number?
Yes, sir.
Page number? And any remaining topics in respiration? Electron transport, okay, clear. Anaerobic reactions, okay, okay, right? And anaerobic reaction, electron transport system. And page number 235. Here given cyanide resistant respiration. Cyanide resistant respiration, I will explain in next class. Plant mitochondrial additional electron transport enzyme. Meantime, just you see the concept of 14.8 cyanide resistant respiration.
Okay, sir.
If you are able to understand about this, I won't explain again. Anyway, inform me in next class. Because this completely, if you understand about electron transport system, it would be easy. Right?
Yes, sir.
So anyway, inform me in next class, I will explain. Right?
Yes,
Next
sir.
in page number 238 for unit 15. Nitrogen metabolism. Nitrogen and sulfur metabolism. First, let's see the nitrogen metabolism. Right? Just wait. Is it clear?
Yes, sir.
Yes,
Right.
sir.
Nitrogen metabolism. Biological nitrogen fixation. Symbiotic nitrogen fixation, asymbiotic nitrogen fixation. In biological nitrogen fixation, there are two types, symbiotic and asymbiotic. And they are given page number 241 clearly. Asymbiotic bacteria, cyanobacteria, symbiotic bacteria example. This is not important in theory, but important in competitive exams. All of you turn on page number 240.
Yes, sir.
Turn on page number 240. Table 15.2.
Important nitrogen fixing organisms.
Yes, important
Yes, sir.
nitrogen fixing organisms. This is not important in theory, but important in competitive. Here given asymbiotic example bacteria, aerobic bacteria, example azotobacter.
Vanilla అండి.
Please be silent. Right. Next, micro aerobic, aspergillum, facultative, klebsiella, non-photosynthetic, clostridium, photosynthetic, rhodospirillum rubrum. These are all belongs to bacteria. And cyanobacteria, unicell Glycopsa, Crococus, filamentous, heterocystus, anabina, Nostoc, filamentous non-heterocystus, Pectonema oscillatoria. And symbiotic bacteria, best example, legume symbiosis, Rhizobium species, and non-legume Frankia, Anabina Nostoc. But here, right, okay. So first, let's see the symbiotic nitrogen fixation bacteria. In symbiotic fixation, nitrogen fixation, the first by
Legume symbiosis.
legumes,
Yes, sir.
nitrogen fixation, symbiotic, nitrogen fixation. The best example already we have learned from the 6th class, legumes, legume plants, right? So there are clearly 4 steps. And first, this is the root structure of legume plant. These are the soil particles. Right. Soil particles. Root hairs. This is epidermis. Epidermal layer. Root hairs. Here rhizobium bacteria. Rhizobium bacteria habitat in soil. Rhizobium bacteria. In first step, where is infection thread? Right, okay. Structure. Right. In first step, this rhizobium grows as a group near of root hair. They are developed as a cluster around the root hairs. This recognition of roots by particular rhizobial stains, by flavonoid compounds and lectins. After formation of clustered manner of rhizobium bacteria, the lectins and flavonoids are produced from the host. Lectins and flavonoids are produced from the root. From these chemicals, the bacteria are attracted. Bacteria attracted and aggregated around the root. In response to the bacteria, root hairs curl at their tips. The next step, this is root hair normal. After recognition of this bacteria, immediately this root hair recognized the rhizobium bacteria, and the next step, this is curved into bend-like structure. The curl at the tips. This is called as Shufford crooks, something, any idea? Scientist name, crooks. Right, Shufford crooks. This bending called as Shefford crux. Shefford crux. The later the bacteria proliferated and passed through the infection thread. Let after bend into this like structure, the infection states are formed. Through this infection thread, this aggregated bacteria enter into the root. First root air entered the root air to the epidermis layer. From epidermis layer, finally settled down at the cortex. After formation cortex here, this rhizobium rapidly increased in cortex cell. That's why this cortex cells also rapidly increased as a mass group of cells. As a result, formation of nodules. Like this, root nodules are formed. In this root nodules, rhizobium bacteria present. This is infection thread. Inside infection thread, let me change color. Bacteria. Number of bacteria are formed. So in which nodules, number of bacteria are formed. Like this, we can see the page number 242, root nodule formation in soybean, in legume plants. And also given clearly diagrams also, page number 243, SFI root air, infection thread formation, after finally nodule formation. Right?
Yes, sir.
Is it clear?
Yes, sir.
Only 4 steps. First, position of bacteria. How it is recognized root air? How it is
By the
enter into the
lectin and the flavonoids.
right? How it is enter into the root air through epidermis, finally cortex. What happen in cortex cells? In cortex cells, here the
Nodules are formed.
nodules are formed.
Yes,
Right.
sir.
So this is only just basic of the nitrogen fixation, symbiotic nitrogen fixation, but not completed yet. To be completed,
Okay, sir.
mechanism of nitrogen fixation and mechanism of knot genes, mechanism of leg hemoglobin formation. These are all to be completed. So this is only just concept of symbiotic nodule formation, symbiotic nitrogen fixation. And another one is in heterosis nitrogen fixation, also important. That is in cyanobacterial symbiosis. Next one, symbiotic nitrogen fixation. Second, cyanobacterial symbiotic nitrogen fixation and heterosis nitrogen fixation. These are all to be completed. Okay, clear?
Okay, sir.
Right? Any doubt in this?
No, sir.
First of all, electron transport.
No,
Transport
sir.
system, oxidative phosphorylation. How can you define oxidative phosphorylation? Actually, I did not give any definition, but can you give oxidative phosphorylation?
Where the formation of ATP is going to have. Sir.
How ATPs are formed? Okay, right. ATP formation, phosphorylation, right?
Yes, sir.
What are involved in formation?
Food is oxidized and the energies get produced.
By oxidation process. Simple, the ATP formation by oxidation. By
Yes, sir.
oxidation, what year?
Phosphorylation.
No, here oxidation of which are undergoes to
Na-
oxidation process.
NADH2 and FADH2.
Right, NADH2, FADH2, right. It's clear?
Yes, sir.
Any doubt today class?
No, sir.
Right, and I will explain later in cyanide resistance respiration. అది మీరు కావాలంటేనే చదవండి, ఒకవేళ మీకు అర్థమైతే నో ప్రాబ్లం. ఆక్సిడేటివ్ ఫాస్ఫోరిలేషన్ లా దాంతో కంపేర్ చేసి చదవండి. జస్ట్ కంపేర్ విత్ ఆక్సిడేటివ్ ఫాస్ఫోరిలేషన్. వాట్ ఆర్ ద డిఫరెన్స్ బిట్వీన్ నార్మల్ ఆక్సిడేటివ్ ఫాస్ఫోరిలేషన్ అండ్ cyanide resistant? Simple, here the skip complex 3 and 4 and some slightly changes are there. Hello?
Sir.
Hello?
Yes, sir. Yes, sir.
మీకే ఎందుకు ఇస్తున్నంటే ఇది మీకు ఆక్సిడేటివ్ ఫాస్ఫోరిలేషన్ క్లియర్ గా అర్థమైంది లేదు తెలవాలంటే ఇది మీకు హోంవర్క్ అనుకోండి.
Okay,
Okay,
sir.
sir.
కంపేర్ చేసుకోండి. నువ్వు కంపేర్ చేసండి, ఇది వస్తుంది అనేది నేను కాదు అనను. ఇది ఒకవేళ రాకపోయినా అదైతే ఖచ్చితంగా నేర్చుకుంటారు కదా, ఆల్రెడీ తెలుసు కాబట్టి.
Okay, sir.
Okay, sir.
తర్వాత దీన్ని డిస్కస్ చేద్దాం.
Okay, sir.
నెక్స్ట్ ఎనీవన్ హియర్ ఇంకేమైనా స్కిప్ అయి ఉంటే చెప్పండి. రైట్?
Okay, sir.
కెన్ ఐ లీవ్?
Yes, sir.
Yes, sir.
Thank you so much, sir.
Thank you,
Right.
sir.
Thank you, sir.
Before you leave,
Ah.
sir, do we need to come to the university and submit the assignment, sir?
On the time of practical examination, practical training classes, you will have to submit.
No,
When
sir,
are
they
we
have
having the
given
practical, sir?
Maybe April.
But the deadline was given as 20th of March, sir.
For submission of assignment.
Yes, sir.
Yes, sir.
If you complete, you can submit.
Otherwise,
You can submit on practical training classes.
okay, sir.
But
Okay.
it is not official. But our convenience, I will give you chance to you.
Okay, sir.
Okay, sir. That is good.
Don't
Sir,
put the
any
message in group.
Okay,
Yes,
sir.
sir.
Okay, sir.
But official date is 20.
20th. Yes, sir.
March, 28th of March. Okay.
But sir, what would be the tentative date for practical, sir?
Practicals, sir.
Maybe April, I think second week.
Mhm.
Second week. Okay, sir.
Okay.
The main theory exams?
July.
July. Okay,
Any tentative, sir, dates in July?
No idea.
Okay, sir. Thank you so much, sir.
Okay, right. All of you, Women's Day wishes to all.
Thank you so
sir.
much,
Thank you,
sir.
sir.
Thank you,
Thank
sir.
you, sir. Thank you, sir. Thank you,
Right.
sir.
Thank you, sir.
ಮಾರಿದ್ದೀವಿ
Bye.